Quantitative Biomedicine and a member of the RCSB PDB staff. She goes by Shuchi, and she's going to introduce the training on molecular visualization using the RCSB PDB MolStar tool. And then we'll move into a series of molecular visualization exercises in your respective groups. Christine, am I correct in thinking that the introduction of the alpha team will take place at the end of the afternoon? Okay, thank you. Okay, thanks again, Steen. We look forward to seeing you on Wednesday.
Thanks, this was fun.
It'll be fun to hear you speak then. Thank you.
All right, see ya.
Shuchi, I think you have the floor.
Yeah. Good afternoon, everyone. This is going to be a much more interactive session. I am going to introduce certain ideas to get us going. And then there is an exercise and activity that I'm hoping all of you can independently participate and go through the exercise. So I'm going to try sharing my screen. Okay. So is everybody able to see the slide? Okay, thank you. So I wanted to start off by saying that we are going to do visualization in this session, but the tool that we are going to use for this is a tool that is relatively new on our website. It is relatively new for me too. I wanted to begin by thanking two of the participants in this group, Tejasvi and Stephen, who helped put together documentation, and we learned and discovered many functions of the tools together. So within the groups, you have people who are actually very experienced, who you should tap into when questions come up or when you have situations which are not easy to display, and we'll talk about some of that. So I wanted to start the discussion by getting to know you a little bit, because I know that there are people from all different different universities, different levels of training are participating. So can we have the first poll? And here I invite all mentors and participants, students, to respond, and any guests who are here today. How familiar are you with the RCSB PDB? So please respond. And we still have a few more folks. So it's appearing that a lot of you have heard about this in a class or course that you have taken at some point in time. And good to have some of my colleagues here who are very familiar with the RCSB PDB. So can you see more people completing it, or should we end the poll?
All but 6, I think you're good.
Okay, so thank you for responding. So I also see that some of the people have just been introduced to the RCSB PDB in the context of this bootcamp. And so the reason I wanted to know this is that I'll take a little bit more time and I'll welcome questions from the group to get to understand what is in the PDB file and how to think through that, so that when you're doing your visualization, you are far more effective. Can we have the next question, which is, what is in the PDB file, to your knowledge? Okay, so I think we'll probably, so most everybody knew that it has sequences, structure, domination details, coordinates, and that's great. If we go to the next poll. And here I'm asking you if you have used any of the molecular visualization tools, which ones have you used? So please select any, if you've used multiple tools, please click all of them, or if you've used only one, or if you've used nothing, that's fine too. You can just,
Shuchi, it looks like only one selection holds here.
Oh, is that right, Ken?
That would be my fault.
Because I can imagine that many of you have used multiple different tools.
I can recreate this one, Shuchi, if you want that to be multiple choice.
Yeah, I would appreciate, because I know for sure that there are many folks who have used multiple tools. But I'm also noticing that, I don't know, maybe somebody did click RASMol and PyMol and only the last one got selected or whatever. So maybe, Ken, if you can recreate it, we'll do it a little bit later in the talk. So if we can we get the pop-up box away? I still see it on my
Your
screen.
co-host, you just click the polling button to get rid of it.
Okay. Okay. So what I hope to do in the introduction is tell you a little bit about navigating the PDB. So the data are coming from the PDB. So the first thing that you might want to get comfortable with, and for those who are already comfortable, please be patient. But since there are some folks who are new to this, where do you find your file? And then we'll go over what is in the PDB file. We saw that it has sequences, 3D coordinates, and various other kinds of information. But what about that PDB file is going to be useful when you're doing your visualization? That's what we'll look at a little bit carefully. And then we'll talk a little bit about visualization in general, and then get into the MoldStar tool and do some demo and exercise. So the PDB, so like you heard this morning, even if you're new to the PDB, it has over 165,000 structures. And so each structure, it has a unique identifier, which is what we call the PDB ID or PDB identifier. And when you search for a particular protein by name, let's say hemoglobin, myoglobin, or the main protease in coronavirus, you'll not just get one structure, you'll get lots of structures. And so it is up to you to select from that result list the structure or structures that you want to explore further. And the entry point into the PDB, there are two entry points. One is our default view. This is our default view, which most of us will probably be using. The PDB 101 is a view that we created for students and educators. This also has a lot of very interesting articles that may be relevant to your activities here at the bootcamp. So I encourage you to go play with the various resources available here. But limiting our view to the default view, the rcsb.org, if you type in the top window here, if you type in a search item, such as COVID-19, it'll start giving you all these different options. So it's asking you, are you looking for COVID-19 in the citation title, in the keyword, in the name of a molecule, and whatever. So this is what you can do. When you get the result list on the left-hand side of your screen, you will get a menu that looks like this, which allows you to refine the search. So when I did the search, whenever I took the screenshot a few days ago, it said it had 249 SARS-CoV-2 related structures, 21 Homo sapien structures. So I could narrow down my search result by organism, by experimental method, by polymer type. If I'm looking for only structures that contain both protein and RNA, then I can quickly narrow that down over here. And then beyond that, I can actually build the query further. So there is an option to refine the query where I can say that the result needs to have COVID-19 and this particular author or this particular, protein name or this particular ligand. And this helps you narrow down your search result from 200 and something to maybe a more manageable number like 30 or 20 or 5. And that you can possibly organize and start looking at, because 200 is way too many to start looking at, okay? And so these are the different query search possibilities. You can search by name, by ID, by author, citation, sequence, annotation, etc. You can also browse. So the entire archive is served up with these browsing organizations. So by the drugs that are present in the structure, by the biological processes that the structures are participating in, enzyme classification number, genome location, molecular function, so on and so forth. So there are many ways, many classifications that are built using these structures, and you can browse through them. So even if you didn't know exactly which structure you're looking at or looking for, you might be able to find a group that meets your particular interest or requirement, and then you can, from there, narrow down the number of files you want to explore. Now, once you have identified a structure that you are interested in, you will land on that structure's structure summary page. This is a page that for every single structure in the PDB entry, PDB file, so every PDB entry has a structure summary page. This is what is contained. I'll show it to you quickly. It gives you a sort of quick glimpse of what this particular structure is all about. So here is the PDB identifier. So this is an old hemoglobin structure from Perutz's work. And there's the structure title. It tells you what it is all about. It gives you a quick snapshot of what this molecule looks like. It tells you a little bit about the quality of the structure. It tells you who solved the structure. Any publications associated with this will be, so if you scroll down the page, the next section will be literature. So in the literature, any publications associated with the structure will be described here, along with links to PubMed, the DOI, everything is available. So if you open this, you can read the abstract right here. And I'm pointing these things out because as you are navigating through different structures, you might want to quickly understand what experiment was done, how did they come to this result or conclusion, so that can be rapidly done this way. This section is something that we will start using even today, where it lists very clearly what proteins or nucleic acid molecules are present in the structure. So these are the macromolecules, okay, polymers of some sort, proteins, nucleic acids, etc. And so in here, we see that there is one entity with 2 copies, chains A and C, another entity with another, again, 2 copies, chains B and D. So the hemoglobin alpha and hemoglobin beta, 2 copies, that is listed here. There are ligands, which is the next section, small molecules or ligands. So this is a hemoglobin structure. There are heme groups, and it also happens to have phosphates. So you might see other structures which have sulfates or other small ions, ligands, things that either are required for the function of that particular molecule, or maybe they were just present in the buffer or cryoprotectant or whatnot. Okay, so that is pretty much what I wanted to tell you about finding the file of your interest that you're going to explore. Now let's talk about the file a little bit. So we said that it has coordinates, it has experimental data and metadata. So the experimental data per se, I'm not going to talk about that much because those are what will calculate the maps, those are what will give you different restraints, etc. I'm going to focus on the coordinate file because coordinates are what you will be visualizing using the visualization tool. And so the coordinates actually look like this. So every single atom in this particular valine, that is the first amino acid in this chain, leucine, the second amino acid in that chain, these are the X, Y, and Z coordinates. So that is what is going to be plotted by whatever tool you use. Today we're going to use Moldstar, but it can be any tool. That's what it's going to plot. And then besides the coordinates, there is also information about who solved the structure, what kinds of proteins or ligands there are. There is also information about the sequence and so on and so forth. So these are some of the things that are contained in the PDB file. Now this is probably not something that you will see much. What you will probably be more interested in seeing is a picture like this. So either you've seen this picture in a textbook or the journal article that you're reading, or you would want to produce a picture like this to add into your lab report, or your research results will be summarized in this way. And so this is where we really want to get at. But behind this is the coordinate file, and I just wanted you to have a quick look at it. This format is actually a legacy format. We have sort of put this aside. It's not gone away, it's still there. It's easy to read. It has a fixed character width. So it has 80 characters across each line, and that's as far as it goes. But that becomes complicated if we consider the really, really large structures that we are receiving for very many virus particles or ribosome structures and things like that. So they do not fit into that file format. And for that, we have transitioned to this new format, not very new though, but this is a more modern, more powerful format. It does not have any restrictions about the number of columns that can be fit in one row, but it's not as easily readable. It is machine readable, but it takes your eye a little bit of practice to get used to the idea. So here I wanted to bring up a couple of vocabulary terms that will come in handy when we look at the visualization. One is called entity. So entity is the type of molecule. So for example, when we were looking at the hemoglobin structure, we saw two entities, the hemoglobin alpha chain and the beta chain. So these are two entities. Notice that there are two copies of each entity, right? So this is something you need to get comfortable. And for a polymeric entity, this is the sequence. And again, we will use the sequence as a mechanism for searching and highlighting and selecting things. So I wanted you to get comfortable with these terms. So when we are looking at the entity, so like I said, there are two green chains. So the two copies of the hemoglobin alpha, two copies of the hemoglobin beta, together they make up this tetrameric structure. And here are all the different amino acids shown in this representation. Now, a couple more vocabulary terms. So polymer is again the polymeric chain of, in this case, there's only protein, but you might encounter in your other explorations, nucleic acids like DNA and RNA, which are also polymers. Then there are ligands. So in here we have the heme groups as ligands. That is what the Moldstar will call this, or you can see it listed under the ligands, or it will be understood as a ligand. And then there are these water molecules that will show up. So these are these little red spheres that kind of show up all over the space, and you can turn them on and off. So many a times these water molecules are just producing or creating a hydration shell, but sometimes these water molecules have key roles to play in either stabilizing interactions or sometimes even participating in the chemistry that was captured in that particular structure. So any questions before I go further? I'm going to pause for a quick moment, and I'm going to look at the participants. So if there is any question, I can take it here and then move forward. So in the chat. Okay, so everybody's cool so far? Will we be exploring PDB files or MMSIF files? So in the visualization exercise, we will just put in the PDB ID. And the tool knows to pick up the MMSIF file, and it will be able to look at all the functionalities or all the details that the MMSIF file has can be explored using the various options. So our focus will be getting familiar with the interface, but in the background, it is reading the MMSIF file. Does that sort of answer your question, Paul? And I just also wanted to add that through the course of the week, when we do the various Pi Rosetta and mutational analysis and whatnot, we might be outputting or PDB format files, which can then be uploaded into Moldstar as necessary for analysis. But you will also be learning how to use some of the analysis options in both Foldit and Pi Rosetta. And Joey, correct me if I'm misspeaking something. Okay.
Here's time from Stephen Mills.
Stephen Mills. Which am I most familiar with?
Sorry, I raised my hand separate from that question. I'm looking at one of those MMSIF files real quick, and it looks like the XY coordinate part of the data is pretty much the same as it was in the old style, and it's mainly the header that's different in the two different formats. Am I about right?
So what is shown, so I'm going back to this, so you're looking at this slide, right?
I pulled up the MMSIF file for the main protease and was just looking at the raw file, but yes,
yeah. So the thing is that each of the columns headings is listed in a loop structure written right above the coordinate section. And so, yeah, it's XYZ. So in this particular file, it looks exactly the same, but there are files where there are additional terms, additional information included. So it doesn't look as clean, it looks a little more complex. But yeah, this information content does not change. In fact, the MMSIF file is far more detailed and has more information. Does that sort of answer your question?
Yep, that's what I need. Thank you.
Okay. So yeah, so you're right. Mike Picard said, what are the aniso lines? So the anisotropic coordinates, so those are, if the file has anisotropic data, those additional details will sort of intersperse between the regular coordinates, and that makes it a little awkward and, you know, not easy to read visually. But given to a computer, it knows exactly where to pick up information from and do visualization or calculation analysis, whatever is necessary. Okay. Any other questions before we get into a little bit of history? Okay, so I'm gonna go back and talking about visualization, so if we go way back to Perutz and Kendrew, they did their visualization in a very physical format. So they didn't have these fancy tools and computers on with these softwares that would allow us to do all this visualization that we'll be doing today. They actually were constructing these either metal structures or wood slabs forming these contour structures. You see Perutz holding this hemoglobin model, and this is a myoglobin model. And it is, to me at least personally, it's very impressive that you know, they were able to construct these from the calculated densities, and they were able to capture coordinates that were meaningful. I mean, yes, if you look at the current validation standards, maybe they don't do very well in those standards, but it's really impressive that they got it all together. But today we have the luxury of these powerful computers and tools that allow us to just, with a click of a button, show these very complex models and then go in and investigate. Now, the idea of investigation and focusing on the business end, or what is interesting, was sort of pioneered by the artist Irving Geis. So Irving Geis actually has made a series of paintings where he's taken a molecule like this. So this is myoglobin, and this is also myoglobin. And you can see that he's taken this really complicated structure and simplified it and allowed us to focus on where the business end is, which is the heme group, that is being held together by these two histidines, and in this case, water. And the whole thing is being held within a cage of 8 helices. Now, it was his ability to sort of remove all the side chain complexity and just focus on the backbone that allowed us to see this sausage-like model and focus in on where the heme is and what is it doing and how is it really able to bind and release oxygen. And along those lines, we have to, of course, mention Jane Richardson, who came up with this whole idea of the ribbon representation. And so no matter which tool, which software you have used, I think all tools have a version of the ribbon representation, which is really a very stylized way of connecting all the C alpha atoms of going all along the polypeptide chain. And it is stylized and connected in smooth ribbons. So in the regions that are alpha helical, it shows up as a helix. In regions that are beta strand-like or sheet-like, you see these long arrow-like structures. So the head of the arrow points the end to seed terminal direction. And so by that scheme, you can take any molecule in the PDB and use the ribbon representation to show which parts of the protein have helices, which have sheets or coils, and so on and so forth. And this is a very common representation that is seen in pretty much all, wherever we see it in textbooks, in the literature, in any kind of media, we see this. And then, of course, behind the development of this whole field of molecular graphics lies the gaming field. And with the powerful technologies and tools and things that they have developed, we can now use some of those technologies to look at these really large, really complex structures, like is shown in this particular here, which is a chromatin fiber model. Now, I wanted to, before we jump into the tool itself, I wanted to quickly talk about some metaphors and conventions that are used in, no matter which tool you use, these conventions still hold. The first view that you're seeing here is the wireframe or stick, ball and stick. It's called in various ways. But what is shown in here is every single atom that is present in the coordinate file is shown as a dot or a ball, and all its covalent connections are shown with a line or a stick. And they are color-coded often. Sometimes the carbons are colored in different ways, but usually the nitrogens and the oxygens, they remain blue and red respectively. Sulfur is yellow most commonly. And this is really giving you everything that is in that PDB file, in that coordinate section, is shown in here. And so it looks a little busy. It does not quite immediately give you a clear understanding of where is the ligand, where is the helix, what is going on over here. It looks like a fuzzball, I have to agree, but it still gives you the sense that this is some kind of a globular protein. That's, I think, the only takeaway that I would take from here. If you move to the next format, this ribbons representation, it immediately gives you a sense of how regions of this particular protein is helical. And there are many helices that are not obvious in this view, but are obvious in here. And the color scheme also very quickly helps you recognize that there are possibly 4 different something. So in this case, there are 4 different chains, and these ball and stick representations show you the positions of the ligands that are bound in here. Now, if you remember, Jane Richardson's assumption was to just chart the C alpha atoms. So she removed all the side chains and any other atoms that are present in the structure. And so it appears that these ligands are kind of floating in space, which is so not true. If you add back the side chains and look at the surface, this is what you will see, where you will see that the ligands are actually snugly fit in these different pockets on all the 4 chains. And that's the heme that is bound to each of the 4 alpha and beta globin chains. And so if you just remember this metaphor that, you can have all atom representations or only backbone representations. You can have ball and stick or surface representations. And the choice of which representation you choose for your particular figure you're making will entirely depend on the story you are trying to say. So this is a figure that was generated by a student many, many years ago. And she was trying to discuss how there were two drugs that bound to two different pockets in the same site in a particular kinase. So the red drug bound in this direction, the green drug bound in that direction. And there was a mutation over here which actually interfered with the binding of both drugs. And so she chose to show surface, ball and stick, ribbon, all the different things that were necessary to communicate her story. So again, to reiterate, depending on what is it that you're making the figure for, you might want to choose one or a combination of different representations to showcase whatever feature you're trying to highlight or whatever analysis you're trying to do. And with that, we are actually going to move into the hands-on exercise. So what we are going to learn to do today is look at the file in the software in Moldstar. We will do a little bit of navigation, selecting chains, displaying the chain in different coloring formats and so on and so forth. And then I think I will stop. I'll do a quick demo to give you a sort of lay of land, and then you will take on the exercise and start working through it. We will try to complete at least the initial piece of exploration of the main APO structure today, and you're welcome to continue working on it as later this afternoon or evening. Tomorrow we will continue that exercise. It will be, tomorrow morning we will start off with an introduction to ligand binding to this particular protease. So that'll give you a little more context to what you're seeing when you look at these inhibitor bound structures. We'll continue that. And then we will actually do some superpositions to see how the APO structure without any ligand or inhibitor compares with the ligand bound structures, and what their differences are, similarities are, things like that. And we'll also, I think, in the end, there is also a comparison between the SARS main protease, the original SARS main protease, and the SARS-CoV-2 main protease. So we'll get a chance to see how similar or different they are. Okay, so I am going to stop sharing this. And while I'm pulling out my browser, if there are any questions, I'm happy to take that before we go into the demo. Any questions? Okay, so let's just get started. I am going to share this screen now. So can everybody see my browser with the PDB homepage? Okay, so as an example, I'm going to use the myoglobin structure 1MBO to give you a quick tour, and we look at the 4-HHP, which has many 4 chains, just so you get a sense of what a monomeric protein and a multimeric protein look like, and then you can go in and start looking at the exercise. So I'm going to go to the myoglobin structure here. And like we described, here are all the different sections. So here's your small molecules. So notice there is, besides heme, there is a sulfate group and oxygen. And there is only one single protein chain, as this is myoglobin. So just by clicking on this 3D view, you actually open a page where the coordinates of this file are displayed in Moldstar. So just to get you introduced to this particular page, This is your 3D canvas. This is where the molecule is going to be displayed. This is your sequence panel. So you can select any of the specific amino acids here or here. And notice if I highlight this, the corresponding one gets highlighted in the sequence. This is your control panel. The right-hand side is your control panel. This is where you will see various options that you can apply. This is also a series of buttons that will come in very handy. So the file is automatically loaded for you as we are looking at it from the structure summary view page. You can take a quick snapshot. You can click on this and it'll ask you, you can just download it. So I just wanted to get this out of the way first. Another thing I wanted to say is that if you want to increase the working space for this particular tool, you can just click on this and it opens it up. I'm going to keep it closed because the Zoom participants things are showing up and that's kind of confused me. But okay, so quickly, so the mouse control, it's very intuitive. There's really not much to tell you specifically, but if you still have a different kind of device and you are finding it uncomfortable, there is help documentation here. So you can click it open. This gives you very detailed information about all these different things. So feel free to play with it. Here, I wanted you to draw your attention to this components section. So remember we talked about polymer, ligand, and water. So these are all the waters which I can turn off and on. The ligands I can again turn off and on, and the polymer off and on. There are many other options that can be activated by this 3-dot options. So here in the polymer, I can say I want to change the coloring. Instead of chain property, I want to change the coloring to residue property sequence ID. So this is something that a lot of you may be familiar with. It's called the rainbow color scheme. It tells you where the chain begins and where the chain ends. So this is the N-terminal end. And notice as I'm mousing over, at the bottom of the screen, it is telling me that this is valine 1. And I can go to the red end, and that is glycine 153. So that is the C-terminal end. I can mouse on any particular location on any of the helices, and it's telling me the position. This is aspartate 60, and so on. Okay. Now, so one of the things I like to do is, since I see that there is a ligand, I want to see which amino acid residues are holding that ligand in place. How are the interactions stabilizing the ligand? Is there some covalent linkage, non-covalent linkage? What is happening there? And so if I just click on that, it focuses, zooms in, and it also draws all these different non-covalent interactions. Again, if I mouse over any one of those, if I mouse over any one of these interaction lines, it'll tell me that this is a hydrogen bond between those two atoms. This is sorry. So if I click on it again, it goes back. So this, for example, is the heme, but this particular interaction is a coordinate. Why is it not showing up? So let's try this. So this is a metal coordination between the histidine. This is the proximal histidine to the iron in the heme. And this is where the oxygen is bound. So you can play with this. You can learn a lot. And you can actually add or change the types of interactions that you are able to see. So you can expand on this, and in the covalent, non-covalent interactions list, you can say, okay, also show me ionic interactions or hydrophobic interactions, and then click on it to see what happens. So you see additional colored lines have shown up. So I will let you play with all this. So this is if I can clearly see what, the ligand is or which amino acid I want to click on. But what if I'm reading a paper and in the paper it says, residue number 60 has some interesting interaction. So how would I get to that? So for that, I think using the sequence panel makes it easier. So I can just click on residue number 60, and it's showing up as ASP, and then it's showing all the various interactions. But if I wanted to select a whole stretch and maybe hide it or color it differently or whatnot, then I think it is easier to, and notice if I click anywhere outside in the white space, it deselects it, it gets away from it. So here's the tool for selections. So this selection allows you to select something. And when I say select, this is really informing the software that I am thinking about this particular residue or this particular chain or atom or entity or whatever. You can have the picking level set to whichever one is appropriate for you. In this case, we are keeping it at residue. And let's say I want to select the region 61 through 71. Okay. Notice it got highlighted in this green color, both in the 3D canvas and in the sequence panel. This is the set that, or the amino acids that the software is now thinking about, and whatever instructions I give it, I can say hide it or make it go away, it's gone. I can undo that. I can also say color it magenta, because that is interesting or has some, oops. And it didn't do it. No, it did do it. Why didn't it do it?
I don't think you had it selected.
It didn't select it? Okay. So let's do 61 through 71. It is selected. Oh, because it has the green color, it fooled me. So sorry, I want to color it magenta and apply. And so here I have colored it in a different color. Now, one thing that you might want to do is, let's say you're looking at the active site, and this is something that you will need for your examining your protease structure. Let's say you have a set of amino acids that you want to identify, which are important for catalysis or binding or whatnot. And so here, I'm just going to make that up and say it is, let's say this, I'm just going to make it up as, let's say it's this residue, okay? And that residue. Now, if I want to see the side chains of this, now notice that in our view here, if I do anything to the polymer, it takes away the whole polymer. Whole thing is hidden or taken away or whatever. I just want to see the side chains of these two amino acids. So the way that this tool is written is that you can create a component. And the component is something that the software thinks of as a group of either residues or atoms or chains or whatever you want to say. And so in fact, what you're seeing here, all polymers in the structure they make up one component. All ligands make up one component and so on. So here, I'm going to create a component that comprises just these two amino acids. And I'm going to say, show this in a ball and stick representation. And I'm going to call this interesting, okay? Okay, so now that the interesting component has shown up, it has these side chains shown because that's the way that I wanted it displayed. But now I can also show it as surface. I can, color it in different ways, do all kinds of things. And I can, select and do measurements, analysis, whatnot. So I think for a single polymer chain, I think these are some of the main tools that you might need or want to play with. Other than this, now, of course, this is going to make it very difficult for me to measure anything. So I'm going to go into the interesting component, and the molecular representation or the surface representation, I can hide it, or I can even delete it. But one other function I wanted you to see is measurement. So measurement, if you're doing measurement between two atoms, distance, you need at least 2 atoms. If you're doing an angle, you need at least 3. And dihedrals, you need 4. So let's say I want to measure the distance between these 2 residues. If I did it at the residue level, it'll do a measurement between the centroids of those 2 side chains. But if I change this now to an atom, then I can select that atom and that atom, sorry. And go to measurements. And notice these 2 atoms are selected. I say, show me the distance. And it computed the distance to be 7.26, okay? So I think with this introduction, I feel like, I've sort of introduced the key areas and key functions here. Tejasvi and Stephen, do you think that there is any other function that we should introduce before we take on the exercise?
No,
I think we'll just get started. So
thank you.
the exercise is, if you go to the home page, if you click on today's lecture, it'll take you to a page. And the exercise is right here. So if you you can open this. So this is a PowerPoint. Oh, sorry, this is a Word document. So open it on your own computer, and the instructions are provided. So you can follow the instructions and start typing in your answers. Or if you're saving images, start putting in the images in there. If you want to label any particular, like there are domains that you need to recognize and so on, feel free to use a PowerPoint on your computer or any other graphics tool that you like to use for labeling, etc. Feel free to use that. And let's get going. I am here on chat. I will stop sharing this and then we can take stock periodically, like, after 5-10 minutes, we'll see how far people have gone and if they're running into trouble. Because really the activity is only when you do it will you get a feel for it. So I encourage you to download that exercise and take it away. And if you have questions, feel free to chat. I see that there are some chat things going on. So Mike asked, can you show again how you got there? Can you remind me where the there is? So
the exercise, please. I'm sorry, I'm not keeping
sure.
up with your clicks. If you maybe give a second when you click
sure.
something to keep up, that would help.
No
Mike,
problem.
I also put it in the chat. It's just the left menu, click on mole star visualization. And then the exercise is the second link.